Good day, everyone. Our group will present the post-lab for exercise number 4, titled The Development of the Fruit Fly, Drosophila melanogaster. Drosophila melanogaster, also known as the fruit fly, is a small insect that is commonly found near ripening fruit. It is a sexually dimorphic organism and is an extremely fertile organism. It is a widely used model organism for scientific research, and the study of this organism has provided into eukaryotic genetics and human disease. For this exercise, three main objectives were aimed to be achieved. Number 1 is to explain the Drosophila life cycle. Number 2 is to identify the main developmental stages of Drosophila. Number 3 is to identify the characteristics on why it is considered as a model organism.
This activity was done in three phases. First is the culturing of Drosophila, second is anesthetizing by cooling, and lastly is the egg observations. For culturing, banana pieces were placed inside the bottle to attract the flies. After waiting a couple more hours, the jar was secured with a paper towel. After collection, the fruit flies were observed and documented for at least 10 days to visualize their life cycle. For anesthetizing, the culture was placed in the freezer for about 12 minutes. This makes them immobile and so easier for us to observe their morphology and sort the male and female flies. And then, a video was watched to understand the embryobiology, collection, and harvesting. In this video, the harvested eggs were placed into 50% bleach for 2 to 10 minutes to dissolve the chorion. Then, eggs were rinsed thoroughly with distilled water to make sure the naked embryo was undamaged by bleach.
Here we have the fruit fly life cycle, where we start with the embryo, which is a fertilized egg that is about zero point 0.5 millimeters long and oval in shape. After fertilization, the embryo goes through rapid mitotic division without growth. The zygotic nucleus then undergoes a series of nuclear division and form a syncytial blastoderm. After the 10th division, nuclei moves to the periphery of the syncytial blastoderm. After the 13th, the nuclei becomes individualized, forming the cellular blastoderm. Then it undergoes gastrulation, and after 24 hours, it hatches into a larva. Larvae are white with segmented bodies resembling a worm. They progress through three stages. The first instar, seen on the second picture, for 24 hours, the second instar, seen in the third picture, for another 24 hours, and the third instar, which will be seen on the next slide for 48 hours. Molting occurs between each stages. As mentioned, here is the third instar, where when ready for pupation, leaves its food source and attached to a firm surface such as the side of a vial. Next image shows a pupa. Pupa are immobile and are initially soft and white, but eventually harden and turn brown. Over a period of 4 days, larval tissues degenerate and adult tissues form. Eclusion marks the end of the pupal stage and the flies emerge as adults. 8 hours after eclusion, the adults become sexually receptive and begin to mate, starting the life cycle all over again. Here in this slide, we see the female and male form of a Drosophila. Drosophila are also sexually dimorphic organisms, meaning that the females are phenotypically distinct from males. Males are much smaller than females, and they have darkly colored external genitalia as well as more black pigment on the lower abdomens. Males also have a patch of bristles on their forelegs called the sex combs, used to latch onto the female during copulation. Stages in embryonic development of Drosophila starts with early cleavage, where the nuclei cleave in the central region. Next is the migration of cleavage nuclei, where nuclei migrate to the periphery. Then, formation of syncytial blastoderm follows, where pole cells form posteriorly. Next is the formation of cellular blastoderm, where cell membranes form between the nuclei. And then early gastrulation occurs, where the ventral furrow forms, as well as the thickening of the posterior plate below the pole cells. Next is the midgut invagination, where the midgut invagination can be seen ventrally, as can the cephalic furrow. To continue, development proceeds with germ band extension, where invagination of the hindgut can be seen dorsally, and then the stomidial invagination, where invagination of the stomidium can be seen ventrally, and then shortening of the germ band, where hindgut and foregut invagination are deep. Next is the shortened embryo, where hindgut is now fully posterior. Consequently, the dorsal closure, where the ectoderm closes dorsally, the midgut broadens and the head involutes. Lastly, the condensation of ventral nervous system, where the gut regions are joined and then the nervous system forms ventrally.
The cleavage pattern of fruit flies and sea urchin was discussed. Fruit flies have a meroblastic superficial cleavage, while sea urchins have a radial holoblastic cleavage. For fruit flies, yolk that was previously found in the center now only does cleavage on the egg's cytoplasmic rim. Furthermore, meroblastic superficial cleavage only occurs after nuclei division. In sea urchins, the cleavage has 4 stages: the first stage, or the meridional, and second cleavage, or the perpendicular stage, has a cleavage furrow passing through both animal and vegetal poles. The third cleavage, or the equatorial, separates animal and vegetal hemispheres from one another. The last stage creates an animal tier, which becomes the mesomeres, and a vegetal tier, which becomes the macromeres and micromeres. To distinguish male and female Drosophila, we can look at their sizes. Females are generally bigger, while males are smaller. In terms of genitalia color, males have dark pigments on their entire lower abdomen, while females only have pigments on their posterior halves. Specifically, males have sex combs on their forelegs for copulation with females. Conversely, females have no sex combs at all. Fruit flies increase in size during the larval stages and are heavily reliant on the amount of protein that they consume. Maggots actively eat food, for example bacteria and microbes, found in decomposing organic material, for example yeast and fruit sugars. These are used to support their growth over 4 days during the larval period.
In this exercise, we observe the organism Drosophila melanogaster, or the fruit fly. We observed this holometabolous insect through its growth stages. Drosophila growth progresses through 4 main stages of development: the embryo, larva, pupa, and adult. Furthermore, in this exercise, we have observed the sexual dimorphism of Drosophila. We also highlighted the distinction of both sexes. Finally, Drosophila has been widely studied because it is easy to maintain and breed in a laboratory. Additionally, they are easy to study because their genetics are simple in comparison to mammals. This makes it Drosophila as a model organism and makes it perfect for activities such as this exercise.